So good morning. Most of us in the field of orthopedic surgery recognize that there are a finite number of individuals that have advanced our field and are worthy of being considered for a Nobel Prize for truly have innovated. It is rather humbling for me to be able to say that I have the distinct pleasure of introducing and have spent time with our visiting professor. Dr. Kaplan trained at in Illinois Institute of Technology, had his Ph.D. at Johns Hopkins University, and actually started as a professor actually before I was born at Case Western University. And so when I say that he has started the field, it is no exaggeration. Throughout his career, he has published over 400 now close to 500 publications, and has won numerous awards. On a bit of a personal level, two of his awards we share. He's won the Kappa Delta Award as well as the Marshall Ewers Award. And the work I did to receive the Kappa Delta Award, the personal aspect, is when long before I was a surgeon scientist, I was just a technician. I learned how to do tissue culture from our CEO. And our CEO taught me how to do tissue culture, and we did Schwann cell co-culture work. And that was the foundation of the Kappa Delta Award that I won 17 years after Dr. Kaplan did. Dr. Kaplan has won every major award in the field of musculoskeletal tissue engineering. He has won numerous lifetime awards, including the first lifetime award from Terminus. Without a doubt, you cannot talk about mesenchymal stem cells, adipose-derived stem cells, without an appreciation of Dr. Kaplan's work. And it is truly humbling and a distinct pleasure to invite Dr. Kaplan to the podium and share with us some of his insights and knowledge. But before I say that, it has been truly remarkable to realize for someone who's accomplished as much as he has reminded me how very important family is to him and how much it should matter to all of us who continue to strive to advance the field. And with that, I'd like us to put our hands together and welcome Dr. Kaplan to the podium.
So I'm going to stay down at the ground level because I'm going to walk around and make sure that the people in the back are not sleeping or worse. I should make you all check your phones at the door, but I know there'll be some emails coming from the back, so I'm going to watch every move you're making. So I want to start this lecture by thanking thank you, Habib, for the wonderful invitation to come and join you. And I do so because this field is going to explode, and it will require companies to form that provide product for practitioners. And although basic researchers like myself can try to figure out how things work, the real delivery of healthcare is only done if somebody's making a buck. And that's the way our society works, and that's an important aspect. And so I think companies like this are going to be important in the translation of the kinds of things that I do into real medical practice. And I think that's a key element. I'm going to talk about the basic science. I'm going to do it in very general terms, but I'm going to try to take you through the logic for why I'm going to beg you, and I do that with difficulty, beg you to stop calling these cells stem cells, because they are not stem cells. So if this works, so this is my disclosure. I started a company called Invitrx Therapeutics. I don't have anything to do with Osiris, but the university gets a royalty, which they generously share with me, which won't pay for the coffee in the back, so you don't have to worry about me retiring on my royalties. I'm also a consultant for a company called Lipids. You'll see why that's important in a minute. So I always like to be a little controversial. So young women have monthly bleeds, but never get sepsis. I can assure you that the uterus is not sterile. So why, if I got that kind of bleed on my arm, I'd be running to the ER. I wouldn't worry about scar formation. I'd be worried about getting an infection. So why do women not get sepsis? I'll answer that as we go along. So in 1991, I wrote this article, was published. This was the Kappa Delta paper, actually, in which I coined the term mesenchymal stem cells. I'm going to tell you why I did it, and I feel quite justified for having called them mesenchymal stem cells, which is, again, it only took 20 years to convince a few orthopedic surgeons that maybe there were some stem cells in marrow, but we'll get to that later. But in 2010, I changed the name to medicinal signaling cells. Of course, I have this delicate ego, so I want everybody to continue to call the cells MSCs, but they're not stem cells, and that's why I changed the name. So I'm going to give you, the guys in the back, hello guys, I'm going to give you the conclusions so you can do your emails. So the conclusions are that MSCs are derived from perivascular cells, pericytes. The MSCs do not arise from stroma. Stroma is an old histological term for connective tissue. MSCs do not come from connective tissue. They come from perivascular cells. MSCs do not differentiate into cartilage or bone or tendon or muscle in your body. They don't do that. So therefore, they're not stem cells. And what MSCs do naturally in your body is they make drugs at sites of injury. Unique, self-contained, regenerative system that I'll go through. They do this by making molecules which interface with the immune system. In some cases, immunosuppressive, in some cases, immunostimulatory. I'll tell you the difference. They make molecules that set up a regenerative microenvironment at sites of injury. Inhibit scar formation. Set up regeneration. They make molecules that affect pain by sitting on opioid receptors. And also they make molecules that are antibiotic proteins that kill bacteria. And I'll show you the data for all of that. So in the beginning, I put MSCs in culture, and I got them to differentiate in culture into cartilage or bone or tendon or muscle. So what's clear is I can make cells dance on a petri dish to my tune. Mesenchymal cells are highly plastic and influenced by what they see. The dogma in the late 80s when I started this was that what you saw in tissue culture is what happens in the body. Wrong. That's wrong. These cells are not stem cells. More importantly, they manage your innate capacity to regenerate tissue. Or every person in this room is alive because you continually regenerate every tissue in your body. Your skin turns over every couple of weeks. You keep washing those dead cells off because they stink. But if they didn't stink, you wouldn't be using so much soap. So MSCs function to manage the innate capacity for tissues to regenerate. And the example that I always use is the blood system. So in the blood system, every single second, 15 million cells are dropping dead in your body and are perfectly replaced. No, nobody raised their hand and said, I'm super person. I just regenerated 200 million cells while you said that sentence. You do this naturally. And the blood system was thought to be the unusual system, and the blood system has a stem cell in your bone marrow called the hematopoietic stem cell, and that cell, if you eliminate it, you're dead, because all of those circulating blood cells that arise from this hematopoietic stem cell have half-lives, which means they die on time. Some within 20 minutes, some 50 years, stick around for 50 years. No hematopoietic stem cell, you're gone. That's why the hematologists do bone marrow transplants after they've given all these poisons to cancer patients. So in bone marrow, as somebody involved in orthopedics, in bone marrow there was another cell, which I named mesenchymal stem cell, and in the late 80s, I drew this diagram as a hypothesis slide. To say that I got pushback is an understatement, because on a grant where I had this diagram, the reviewer started the summary statement for the NIH grant by saying, it is well known. As soon as you see that sentence, it's well known, you're dead. So it is well known, that means the dogma of the day is that there's only one stem cell in your body, in adult bodies, and that's the hematopoietic stem cell. So by me calling that a mesenchymal stem cell, I'm dead in the water immediately. And the dogma, for example, is that in your heart, you're born with all of the cardiac myocytes. They get bigger or smaller, but they don't increase in number. That's the dogma in 1980s. That's wrong. That's wrong, and I'll show you why. So if you want to do tissue engineering, what you have to do with the mesenchymal stem cell is to bring it down this lineage of differentiation until you get a chondrocyte. Ear cartilage, nose cartilage, throat cartilage, sternal cartilage, hip cartilage, knee cartilage, ankle cartilage, all different. You give me an H&E of any of those tissues, and I can tell you what cartilage. Cartilage is the family name. Ankle cartilage is the specific name. Knee cartilage, hip cartilage, huge differences. So if you want to differentiate something into cartilage, you have to have a site-specific phenotype. That's why nobody's selling tissue-engineered knee cartilage on the market, because we can't do that with MSCs. We're working on it. I can tell you we know some secrets, so eventually we are going to be able to do it, but not today. There's no product on the market for cartilage, articular cartilage. So tissue engineering, you have to know lineages and how to get the cell into the right lineage. I'm not going to talk about tissue engineering. I'm going to talk about regenerative medicine, which only has to do with the MSC. So every tissue that you can imagine, and I've listed on the left side of the slide, there's a paper that says you can get MSCs from those tissues. For me, orthopedics, I work with marrow, so I can't think of anything more revolting than somebody publishing a paper to say that you get MSCs from fat. That's disgusting. How can an orthopedic surgeon even care about fat, right? So, but you get MSCs from fat. As a matter of fact, 300 to 500-fold more MSCs per cc of fat than cc of marrow. 300 to 500 fold. So what's missing from this slide? All those tissues, we can get the MSCs, and we get those MSCs in vitro to make cartilage, bone, or fat, or whatever. What's missing from the slide are blood vessels. All of those tissues have blood vessels, and that's what we missed in the beginning of these studies. So I'm going to go to the end result, and that MSCs come from perivascular cells. So when Habib took his blood pressure medication this morning, those little red cells, pericytes, on the capillary, that's a capillary in heart, those little red cells squeeze the blood vessel to give you high blood pressure or relax to give you low blood pressure. They are the contractile elements of your vascular system. You have 50 to 100,000 miles of blood vessels in your body. Every single blood vessel, without exception, there are no exceptions, has perivascular cells on it. These are scanning EMs of blood vessels in different tissues. Every cell you see is a pericyte. These are blood vessels in tissues. Every cell you see, without exception, is a pericyte. Every cell you see is a pericyte. You getting bored? Every cell you see is a pericyte. Every cell you see, somebody see a tissue that they work with? How clever have I been? I haven't told you that every frame of those scanning EMs is a different tissue. So the pericytes look different in every different tissue. Why? Because they have their backs to that other tissue. Why should they all look the same? Do they all have different chemistries? They all have different chemistries, but they all give rise to MSCs. And so what I know how to do is kick the MSCs off those blood vessels with enzymes or other tricks, put them in culture, and I can make them dance. It's not what they do in the body. It's not what they do in the body. So MSCs in vitro are multipotent, but not in vivo. They're not stem cells. So this is an important paper. You need to take pictures of this, and you need to read this paper. The paper has two important panels. This one, a labeled pericyte on a blood vessel. You put it in culture, MSCs, and we can get all these different tissues. If you take that labeled pericyte and you have an animal that's aging, or an animal that's on high-fat diet and making a gigantic amount of fat, or you break the animal's leg and it makes new bone, the label on the pericyte never appears in the newly differentiated product. So therefore, in vivo, those pericytes don't differentiate into mesenchymal tissues. That's what this paper says. Has some problems in this paper, but that's the conclusion. There's another piece that I'll come back to that you'll see as part of the thesis that I'm trying to lay out for you, which is all those tissues. All those tissues, each tissue has its own unique stem cell. So in heart, the little yellow balls, the stem cell, that stem cell is sitting on an MSC pericyte, and both that stem cell and that pericyte are sitting on a blood vessel, endothelial cells. Heart stem cell, liver stem cell, they're called progenitors, but we're going to call them stem cells because the heart stem cell, stimulated to divide, makes new cardiac myocytes. It doesn't make skeletal tissue. It only makes cardiac myocytes. A skeletal stem cell, which is called a satellite cell, it divides and makes skeletal multinucleated myotubes, different than heart. So every tissue of your body, without exception, has its own progenitor. Your skin, remember your skin? They're called keratinocyte stem cells. They divide like crazy, and that's why you got a bunch of dead cells on the top of your skin, because they replace the ones that are dropping dead. So this is an important aspect. And the important aspect is the stem cell niche always has an MSC pericyte that's making special molecules for that stem cell. So that's another function of MSC progenitors, is that they nurture tissue-specific stem cells. So again, injury, blood vessel breaks, so make off, this is a blood vessel, pericyte, blood vessel breaks, pericyte comes off, and that cell now differentiates, changes into an MSC. From the front of the MSC is a curtain of molecules that stop your over-aggressive immune system from interrogating the injured tissue, because in the injured tissue you have new antigens, and your immune system will go after those new antigens. So this curtain is your first line of defense against autoimmune diseases from starting. From the back of the MSC, so the MSC gets stimulated. From the back of the MSC, it makes molecules that I call trophic. Those molecules are anti-scarring, angiogenic, and those molecules specifically cause the tissue-specific stem cell to divide and make new progeny. That's how you get regeneration. So back of the MSC, regeneration, front of the MSC, control of the immune system. We know the molecules in the curtain. So if, for example, I took some marrow from Habib and I got his MSCs from his marrow and got them to divide in culture and put them into my blood system, of course I would tingle with the thrill of having his cells circulating around my body. But his curtain is so strong that my immune system would not see his cell. So you can use an allogeneic cell because of the curtain. Eventually the immune system catches up. So his cells are not immunoprivileged in my body, they're immunoevasive. The curtain helps the cell evade the interrogation by the immune cells. Immunoprivileged and immunoevasive are hugely different activities. So again, allogeneic cells, if you have an autoimmune disease, maybe your curtain isn't working. We have data to support that idea. So that's the reason for using allogeneic cells. So, so this is a website called clinicaltrials.gov. If you go to that website and you put mesenchymal stem cells in their search engine, you'll see 1,000 clinical trials are now listed. About 370 are active. This is a map of the world. This is a map of the United States. I'm hugely politically active. I hate red states. That's a political statement. I love this map. I love red states on this map because they have the highest number of clinical trials using MSCs. California continues to fight Texas and Florida, but look at this weird cell, way at this weird red one up, it's called Minnesota. Who in their right mind would go to Minnesota to get MSCs? Oh, that's called Mayo Clinic. Ah, Mayo Clinic. They have an MSC institute, and they've been doing this for 10 years, and so therefore they have a lot of clinical trials. This is Europe. Turns out Spain is the leader quite reasonably because there's lots of MSC stuff going on in Spain. So here are all the clinical symptoms in those. Try just copy pasted. Then this is an eye test for the guys in the back. Can't read that, can you? Well, I just shut this off. So now I put it at higher font so that you could read it. And you need to be impressed by the spectrum of clinical symptoms: graft versus host disease, Crohn's disease, ALS, MS, kidney transplant. So 2010 JAMA paper, first authors 10, T-A-N, 159 allo kidneys transplanted into patients. A third immunosuppressed, hugely immunosuppressed, IL-2 immunosuppressed. A third lightly IL-2 immunosuppressed. They have problems. Another third lightly IL-2 immunosuppressed, but autologous, marrow-derived, culture-expanded MSCs are given to these patients. Beautiful protocol which we all can learn from and should think about. When you put a new kidney in somebody, you put a tourniquet on and then you connect all these blood vessels. You release the tourniquet and let the new kidney be perfused. That's when you give the first shot of MSCs. They go to all of these suture sites. They go to the damaged kidney that's been transplanted and do wonders. And then the smart thing they did is 2 weeks later, they give another shot of those MSCs. 1 year outcomes, perfect kidney function, no rejections, 0 rejections, perfect, no infections, perfect everything. They're now doing, the Chinese are doing a clinical trial, 500 patients, no immunosuppression, just using MSCs as chaperones for kidney transplant. 2 shots. One for acute problems and one for problems that still resist. 2 shots, not 1 shot, 2 shots. Very important paper to learn from. So MSCs are not from stroma. If you think stroma, you got the wrong logic. If you think pericytes, it just changes the way you think about how these cells are managed naturally inside your body. So I've changed the name because MSCs function at sites of damage, and because they make drugs, I'm calling them medicinal signaling cells. Medicinal signaling cells. I prefer that you call them Kaplan cells, but I can't get my papers published when I try that because of my snobby colleagues. So I call them medicinal signaling cells. But the easier thing is just call them MSCs. They're not stem cells. Don't take this stem cell nomenclature out of your brain. It doesn't work. There are no stem cell products currently on the market. MSCs dock at sites of injury, broken blood vessels. That's where they do business. They're immunomodulatory or regenerative, and they function naturally to manage your innate regenerative capabilities at each tissue site. So there's one aspect which is completely missed by everybody, and that's that the MSCs are sensory. What's that mean? So they're sensory because they touch other cells and they communicate with other cells and they survey the microenvironment. So here's a paper that explains that exactly. So the same blue MSC, on the left side, you give it inflammatory signals and it makes anti-inflammatory molecules. On the right side, you give it bacterial signals and it makes inflammatory molecules. Inflammatory means the immune system and the hemopoietic system are going to come and take care of those damn bacteria. If you make anti-inflammatory molecules on the right-hand side, you're dead. You'll be infected. You'll never get rid of that infection. Same cell, different signals, and they have a different rollout for what they sense at each of the sites that they dock and function at. Hugely important concept. So I'm going to do one other important concept, pain management. So this is a paper where they make a defect in the spinal cord, painful, to the animal. And they give it human MSCs from marrow compared to fat. And what they show is that those two preparations are equally potent. The rollout of the response of the MSC is determined by the site at which they dock. An MSC from fat and MSC from marrow are equally potent with regard to this defect in this animal. Here, nobody raised their hand back there and said, human MSC in a rodent. We were told you could never do that. The curtain, Habib's curtain, works in rodents. So the immune system of the animal cannot see the antigens on the cells. So human cells, all of the animal models we use in my lab, rodent models, we use human MSCs. Human. And when the FDA comes to me and says, we need to have syngeneic or autologous MSCs from that animal, I say, no, thank you. I have a real clinical model. I have a rodent model, and I'm giving them human MSCs, and I can use those human MSCs for my clinical trial. It's called a real clinical model. The FDA doesn't like it, but we're starting to see them change. So now, for me, a clinical model for human MSCs, put the human cells in the animal. If it works, it's fine. It's a release criteria. So everybody bumps into the wall, hurts like hell. 2 days later, you got a black and blue mark. Black and blue mark. You broke a blood vessel. So it hurts like hell for about 10 or 15 minutes, and then it stops hurting. Why? Because the MSCs saw the screaming nerves and made molecules that sit on opioid receptors. Nothing's fixed in that site. There's no regeneration in that site. The nerves are quieted down because the opioid receptors on the nerve endings are saturated. Everyone in the room, it's happened to everybody in the room. You never asked yourself, why did the pain go away? You said, I'm super person, the pain went away. Mind over matter, forget about mind over matter, opioid receptors. And I'll show you other data to prove this. So this is my good friend, Dr. Cheng, who's at the Cleveland Clinic. He's the head of the graduate program in pain management. And he snuck this abstract into his annual meeting of the American Academy of Pain Medicine in 2015. What he did was just fabulous. He took rodents and tolerized them to opioids. You and I, we take opioids for 10 to 16 weeks and then we become tolerant. That's why we pop more pills to try to control the pain, because we've tolerized ourselves. So these animals are tolerized. So then he comes to my lab, he steals some marrow MSCs, human, puts them in the animals, and they lose tolerance. So low-dose opioids work fine. The better experiment is, comes to my lab first, gets the MSCs, puts them in the animals, and then he can never tolerize those animals. Why? Because the opioid receptors have molecules that are sitting on them made by MSCs. So that's the first data to show that that's the case. And here's a fabulous paper that I never read. until recently. So I'll break it down simply. The rodent experiment, they jammed the TMJ. Anybody who's had TMJ pain knows it's killer pain. So you touch the cheek of the rodent and it goes through the roof. If you give them opioids, you can touch it all you want, won't affect it. You give them human MSCs, you can touch the cheek all you want. Now, the pain guys have antagonists. Those are small molecules that kick molecules off of opioid receptors. So if you give them MSCs and you give a peripheral antagonist and you pat the cheek of the rodent, they go through the roof. So that works for the first, this is an experiment goes 22 weeks. So you can get 22 weeks of pain relief from MSCs. What's interesting is the antagonist that works at the peripheral site works for 3 weeks. At week 5, if you give the peripheral antagonist and you touch the cheek of the rodent will just look at you. Now the pain is controlled by what are called mu receptors in your central nervous system. The MSCs haven't gone to your central nervous system. So at week 5, we use an antagonist that kicks the molecules off the mu receptors. And then you touch the cheek of the animal goes through the roof. So now we know that between week 3 and week 5, the control of pain is central. Chronic pain, hello, chronic pain? Chronic pain, central. Chronic pain is not peripheral. So now the MSCs have somehow organized to get those receptors occupied. Now I'll give you a mechanism for that in a few minutes. So here's a normal clinical trial, 10 women who have TMJ problems. They liposuction the ladies, they get what's called stromal vascular fraction, and each of the ladies, they characterize the stromal vascular fraction. About 40% of the cells in the stromal vascular fraction are MSCs by sorting. They inject them directly into the TMJ area, and 7 out of 9 of the women, they're done with pain. Their pain scores go to 1. They stop taking all these pain meds. So this needs to be expanded into a bigger trial, but there's no question from this that humans are sensitive to the pain modification as controlled by MSCs. So here, there's hundreds of papers on MSCs and central and peripheral nerve involvement. I won't go through them. So pain management, MSCs control peripheral pain, MSCs control central nervous system pain. So if you have an osteoarthritic knee and you squirt some MSCs into the osteoarthritic knee, within 3 to 5 days the pain goes away if you're a responder. Nothing's fixed in that knee. Nothing is fixed in the knee. The opioid receptors are occupied. Hugely important. So pain management by MSCs involves molecules that sit on opioid receptors. So MSCs dock at sites of damage. They survey the site, and they make molecules specific to that site, whether they're from fat or whether they're from marrow, doesn't matter. How do you get long-term therapeutic effects? Remember, mu receptors, 5 weeks in a rodent. It ain't an MSC that's crossed the blood-brain barrier. Here's a paper that gives you the answer to that question. This paper shows that when human umbilical cord, MSCs, are given to a rodent, 24 to 72 hours later, they're gone. They're labeled. These MSCs are labeled. What you see is that label is inside a very special class of monocytes, which are called LY6C low monocytes. The monocytes have eaten them. Labels inside the monocyte. When that monocyte has eaten the MSC, the monocyte changes so that its cell surface markers are CD206 and CD163 positive. Those cells, if they bump into a T cell, they cause the T cell to become a regulatory T cells. Regulatory T cells stay in your body for years. They provide long-term immunological control. So I know nobody's going to read this complicated paper, so I did this cockamamie diagram, because I know that all the young people in the audience will memorize the damn diagram. So LY6C low red monocyte eats an MSC. After it eats, I gotta stop doing that. Eats the MSC becomes instructive. I want to make a point, which Habib won't like, but MSC is just a big exosome. It's a full lunch, not just a snack. So if LY6C low eats an exosome or eats an MSC, it becomes instructed. So now that instructed MSC, when it breathes upon a T cell, it converts the T cell into a regulatory T cell. Regulatory T cells control the immune system at sites of damage for years or cross the blood-brain barrier and can affect mu receptors. So MSCs themselves are a starter set. They start a cascade of events that end up in therapeutic outcome. So if you measure therapeutic outcome, and then some guy at the FDA asks you the MOA, every time one of the FDA people does the MOA on me, I blink, because I want to say something really rotten back to that person. Because there is no MOA, we don't have a microscope and a slow motion camera that goes into the site where they're working. So whatever MOA you tell them, make sure that you have a few measurements to prove that that's the case, but all of us in the business know that MOA is bullshit. Don't quote me on that. And please delete that from the video. So again, injury site, long-term therapeutic outcome are from secondary influences. Hugely, hugely important premise. So this is Tracy change in fields. Tracy Bonfield, a brilliant young researcher across the street from my lab in Rainbow Babies and Children's Hospital. Rainbow Babies and Children's Hospital is famous for treating kids who have cystic fibrosis. So in cystic fibrosis, the mutations are in a transmembrane regulatory protein. That transmembrane regulatory protein controls sodium chloride transport across membranes. So these kids end up with gunk, with mucus in their lungs and mucus in their GI tract, and their physiology screwed up. What kills the kids are bacterial infections. The bacterial infections, for example, Pseudomonas, we don't know how to treat Pseudomonas. So if a kid gets massive Pseudomonas, it's lethal, because again, we don't know how to treat Pseudomonas. So what the guys at the Rainbow Babies and Children's did is that they made a knockout mouse. So they took the transmembrane regulatory protein out of the mouse. So they call it a cystic fibrosis knockout mouse. So what we do is we take an agarose bead with a known number of Pseudomonas particles, we jam it into the lung, for example, of a wild type normal animal, gets a huge massive lung infection. A week later, the wild types are completely normal, no problem. They take care of that infection. The Pseudomonas, the cystic fibrosis knockout mouse who's been infected with Pseudomonas, after 7 days, dead as a doornail. Can't handle the infection just like the cystic fibrosis kids. If we give them Habib's human mouse, remember, mouse, we do this, his curtain's so potent, works in mouse, you give those mice, at day 2, they walk away. We spend 6 weeks working on how the immune system has killed a bacteria. Wrong again, Kaplan. Here's the answer. The answer is that every one of the MSCs makes a protein which is called LL37. LL37 is a very potent antibiotic protein that kills bacteria on contact. Every person in this room, without exception, is making LL37 in your mouth. They're called defensins. They've been studied for 30, 40 years by dentists. We know their molecular structure, we know how they work, etc. MSCs bump into a bacteria, they make LL37, hello, goodbye bacteria. So when your pet animal injures their paw, they lick the wound. You think they're just cleaning the wound. They're putting a layer of antibiotic proteins on the wound. Don't you try it, your mouths are filthy. Survival of the fittest, a layer of antibiotic proteins. Your dogs do not go to the ER. LL37. I just gave you the answer for sepsis. Remember, I asked the question? You were dubious. You break a blood vessel. What happens? MSC. MSC bumps into a bacteria. Hello, goodbye, bacteria. This is a page from an Indian newspaper. For a few rupees, they'll collect menstrual flow, isolate the MSCs, and freeze them down for emergency use. I just saw a lady who just went yuck. MSC, here's papers. Here's papers. There's about 100 papers that show that menstrual MSCs are just as good as marrow MSCs. They're just as good as fat-derived MSCs. An MSC is an MSC is an MSC. They're different. Their chemistries are different. I'll show you that in a minute. Their chemistries are different. Their reaction to a microenvironment is hardwired. No sepsis for massive bleeds in the uterus because it's a bleed. MSCs are generated, millions, multi-millions of MSCs in menstrual flow. So MSCs dock at sites of broken blood vessels and help regulate the regeneration of that tissue. Uteruses regenerate within a matter of days. Not because the uterus is regenerative, because it's just been bathed in MSCs. So there's a problem. I'm not going to discuss this. This is complicated. So the commercial companies like Mesoblast use the same commercial production run for graft versus host disease, low back pain, acute myocardial infarct, and diabetes. Those MSCs are not tuned to the disease that they're fighting. Interferon gamma is not going to tune the MSCs for cystic fibrosis. They'll tune it for lung infection, but you gotta tune the MSCs. And the other piece of the equation is Habib's MSCs in my body, my response may not be good to his MSCs. I may need somebody else's. So each of us is a different genotype, and so therefore our MSCs are different. So the question is, which one's best for my problem? So MSCs are not tuned, and it's two criteria. One is for the disease that you're fighting or injury that you're fighting, and second for the response profile that each of us has built into us. So again, for basic researchers like me, I have to learn what the tricks are for tuning. So we started a clinical trial for kids with cystic fibrosis that have massive lung infections. We got 10 donors, got their marrows, put their MSCs in culture, and into each culture, we put Pseudomonas or staph. One of the donors off the charts on how much LL37 he made. Not only that, but the guy moved from Cleveland to Pittsburgh. I had to send a limo to Pittsburgh to get the guy to get him back to get another stick so I could get more marrow for the clinical trial. He also made huge amounts of anti-inflammatory molecules. So I tried to tune the MSCs by choosing the right donor, by imagining what I wanted those cells to do in the body. Is that what happened inside the body? Who knows? So clinical trial is currently going. It's hugely interesting. The responding kids decrease neutrophils and increase classes of monocytes in their bloodstream. So we know they're responding. Not all the kids responded. So again, the object is how to assay for responders. Not so easy. So I'm going to end by talking about fat, because there's some interest in this audience, some small interest in fat. So fat is, for those of us who can't control our intake of caloric material, fat is considered a bad thing. But every adipocyte that's filled with this fat droplet is surrounded by capillaries. That's why you get 300 to 500 fold more MSCs per cc than marrow. Blood vessels, MSCs, blood vessels, MSCs. Are we getting the message? This is a paper you need to read. So every preparation of MSCs from every donor is heterogeneous. And by doing what's called single-cell seq analysis for RNA, you can identify in fat, MSC preparation, 9 different classes of molecules, of cells. Each cell is distinctly different than its neighbors. And so for dumb guys like me, they color code them. So red, green, yellow, whatever. Now, what's brilliant about this paper is every one of the cells, and even within one group of cells, there's unique molecules to these subtypes of MSCs. So they make a probe to the different subtypes. And then they go back, and the genius of this paper is they go back to the tissue, and every one of those different subtypes comes from a different anatomical locus. So what I didn't point out in the first slide on fat tissue is the tissue's not homogeneous. So you'd expect a cell on the outside and a cell on the inside. Its capillaries are going to be different, and therefore its pericytes will be different, and therefore the MSCs that come from those different anatomical sites are different. So hugely important. MSC preparations, I don't care whether it's from my lab or anybody's lab, heterogeneous. Now you tell me which cell is the clinically relevant cell. We don't know. So not only are the cells from marrow and from fat have different chemistries, but within each MSC preparation, there's huge differences. Somewhere between 8 and 12 different separate identifiable cells because they come from different anatomical locations. Bunch of companies, variety of companies making MSCs of a particular kind. I'm going to talk about lipids because I denigrated fat in the beginning. I have to make up for it. And this is a lipoaspirate, and I like this preparation because this rig is unique. So you put the lipoaspirate into this tube, which is filled with saline. This intake port is stainless steel wires which are crisscrossed over each other, so you further disperse the lipoaspirate. Then inside the tube are stainless steel balls, and you make a martini by doing this with the stainless steel balls, and it mechanically knocks the vasculature apart and knocks some MSCs off. And then the genius of this apparatus is this exit port which has 500 micron holes. So you turn the stopcock, you put new fresh saline in, it washes away all of the monocytes, all the broken cells, all of the oil and fat out, and what you get at the top is a ball of live adipocytes, 500 micron ball, and inside are broken blood vessels and MSCs. How do I know? Because I took this ball, that's live adipocytes, and I put it in, what do? I put everything in culture, right? So these are live adipocytes. They have fat droplets in them, so they'll never sit down on the bottom of the plate. They'll float. 5 days later, we look at the bottom of the plate, and what do you see? You see MSCs. They say, okay, I'm done sitting around the middle of this junk. I'm getting out of here. And if you put this preparation in your osteoarthritic knee, it's a slow release vehicle. You put 2 mLs of anything in your knee, you get 50 mLs that wash through that knee. Actually, that's why 2 mLs of saline can be therapeutic. 2 mLs of saline can be therapeutic. The orthopedic surgeons in the room just shuddered. Can't charge for 2 mLs of saline. Yeah, I'm not going to repeat that for the audience. So, 2 mLs of saline, you get 50 mLs by day 3, that deluge is done. Hello, in these balls, the MSC said, okay, I had enough. They come out, they look around, they see all this pain. What do? Make molecules that sit on opioid receptors. So if you're a responsive patient, 3 to 5 days later, pain goes away. Nothing's been fixed. Nothing. So again, MSCs in this preparation can be the factor. There's no data to show that they are. This is a Kaplan hypothesis. But if the pain goes away, I know who's doing it. So again, MSCs are not stem cells. They're not stem cells. They're making molecules for sites of injury. So MSCs are drugstores. A pharmacopoeia for sites of injury, whether it's your knee or your muscle or tendonitis, etc. Now, we know I won't go through the details, but we did this from studying cancers. The MSCs are embedded in the basement membrane surrounding every blood vessel. The molecule that's holding the MSC pericyte to the basement membrane is PDGF-BB. It'll take another hour to explain how we know that. This is a fact. PDGF-BB, you guys call that PRP. You break platelets, it releases a ton of PDGF-BB. You want to know whether you have an active PRP preparation? Do an ELISA for PDGF-BB. There's a cutoff. Above a certain level, it's efficacious. Below a certain level, ain't gonna do crap diddly. So PRP is the explanation for how PDGF-BB is how PRP works. It goes into the synovial lining, inflamed lining, releases the MSCs from all those blood vessels. The MSCs become activated, and hello, you got relief. Not necessarily curative. So again, MSCs, medicinal. So this is my original hypothesis slide. It's wrong. We never asked the proper question for where do MSCs come from. MSCs come from pericytes. Changes the way you think about these cells. So again, MSCs, remember MSCs, it's hugely important to keep that nomenclature because of my delicate ego. They are not stem cells. They are not stem cells. So the conclusions are listed here. MSCs come from perivascular cells. They inhibit scar formation. They modulate the immune system. They're immunoevasive, and so therefore you can use allogeneic MSCs. They manage pain. They secrete molecules which are angiogenic. They secrete molecules which are antibacterial. They make mitogens for tissue-specific progenitors called tissue-specific stem cells. They survey, sense, and respond to the local microenvironment. They don't do things systemically. They all act locally. They secrete pro or anti-inflammatory molecules depending on the microenvironment. They get eaten by classes of monocytes who then cause a cascade of events which can control long-term therapy. And more importantly, MSCs are not stem cells. Take the word stem cells and delete it from your memory bank. So one last thing, this is a poster that Diego Correa and Rodrigo Somoza and I did for Nature. If you go to that website, you can download that poster. It has all of the new information on MSCs. This is a page that you'll see in every textbook on histology. And what you see is this is bone marrow, and in bone marrow, the hematopoietic stem cell gives rise to all of these circulating blood cells. Well, look carefully at that picture, and what you see is the hematopoietic stem cell sitting next to an MSC pericyte, both of them in contact with the blood vessel. So here's the stem cell pericyte blood vessel. And just to take this one step further, we compare the quiescent hematopoietic stem cell with the active hematopoietic stem cell, and both of them are sitting next to MSC pericytes, and those MSC pericytes are making molecules specific for that stem cell. Those MSC pericytes have different cell surface markers. They're different cells. They're doing different things. This is the same bone marrow. So this cell we can sort out from this cell because the cell surface markers are different. Heterogeneity in every MSC preparation. I don't care what the source is. And the other thing you were all taught in medical school is that the blood vessels are controlled by the sympathetic nervous system. That's wrong. It's wrong. It's wrong. Look at the picture. Picture says that the sympathetic neurons are innervating the pericytes. They're the contractile members of that trio. So the MSC pericyte is what's innervated. Endothelial cells don't contract. So your blood vessels are contracted on the outside, not by the endothelial cells. I'm not going to talk about this. Again, my prediction is that as these phase 3 clinical trials come to completion, there will be acceptance of cell-based therapy for a variety of indications. Within the next 5 years, there'll be at least 10 indications that are approved by the FDA. So some guy in rural California gets a heart attack. Helicopter comes and brings him to San Diego, okay? 10 years from now, that guy's gonna get into his car, go a mile down the road to the urgent care center, take a bag of Habib's allogeneic marrow MSCs out of the freezer, hook them to an IV line, and that will be the treatment. There's a phase 3 trial by Mesoblast that's going to prove that and be approved. It's going to change the way medicine's practiced. Is our MSCs single drug agents? No. Are they multi-drug delivery vehicles to sites of damage? Yes. What's the MOA? It works. We teach a course every spring at Case Western Reserve. You're all invited to come. I invite a whole bunch of other people beside myself to talk about cardiac stem cells, neural stem cells, hematopoietic stem cells, and on the second day in the afternoon, we do a how-to group of lectures. We show you how to make MSCs, and then we take you to a lab and make you do some of the assays for quality control of MSC and show you what the assays are that we've developed in an academic setting. This is my favorite quote. This says that cell-based therapy is finally at the end of the beginning. I wrote the patents for MSCs in 1990. Kaplan rule number 1 at the business school is takes 20 years from the time something becomes public till it comes to market. First MSC product approved in Australia, 2010. You think your fancy cell phones are the hottest, newest inventions? In World War II, they were called walkie-talkies. Big, bulky things. The transmission concepts were the same. They had to have a big, long antenna. You got this tower that you can see every time you look towards the horizon. That's your antenna. So again, fancy phones, better batteries, more microelectronics. Concept was used in 1940s. So again, we're at the end of the beginning. All the research in my lab's generously supported by your tax dollars. And there's a large group of people at the Skeletal Research Center who are actually doing the work. I'm clearly only the mouthpiece. Thanks.
Well, thanks, Dr. Kaplan. You've just made me completely obsolete as an orthopedic surgeon. So we have a few minutes for questions, and I'm going to give the mic to Dr. Kaplan first.
Thank you so much, Dr. Kaplan. Great presentation. My first question, are you selling any of Habib's cells? Because I want some of those too.
Well, as you might imagine, Habib will charge you an arm and a leg.
So I have two questions, one easy, one a little more complicated. With the WIT article, with the MSCs, what's the half-life of these MSCs once we infuse them?
Well, that's the issue. The issue is that MSCs, there's 10 other papers I can quote that show MSCs stick around for very short times. So the logic was that MSCs are hit and run artists. They hit, they do something, and then they're gone. In all likelihood, the immune system eventually catches up with it. And there's some data to substantiate that. So MSCs are very active for a very short time. They set up a cascade of regenerative and reparative events, but then they're not involved.
My other question, as a clinician, is if we are going to implement these therapeutic interventions or strategies, the whole concept of tuning, any thoughts about suggestions how we figure that out?
The whole concept of?
Of the tuning, the MSC tuning that you're mentioning?
Yeah, I mean, tuning, so for my cells in your body, I need two things. I need our genomes sequenced, and I need our post-translational modifications known. So it ain't going to happen tomorrow. But in the end, that becomes precision medicine. So that if you take a pill, your reactivity to that pill from your genome will be statistically understood. It's all a question of mathematical treatment. But in the end, what we're trying to do now with tuning, the response profile of the patient can be assayed. And I'll give you an example. In the published studies for hyaluronic acid injections into osteoarthritic knees, I won't go through all the data, but there's two pieces of information. One is that about 30% of the patients are responders. In responders, 2 mLs of saline will give you 4 to 6 weeks of pain relief. That's an assay. That's an assay for a responder. So in the orthopods that have to deal with me, they actually give a patient 2 mLs of saline and see if they respond before they do 3 injections of HA. So that's a response assay. On the other hand, the tuning of the MSCs, I gave you the example for the cystic fibrosis kids where we tried to pick the right donor. But eventually, we have to become more sophisticated than those two assays. But that's an example of response tuning. That's going to be some kind of assay. And optimization of the MSC for the ailment that we're using it for.
Just so I can get many questions in, I'm going to limit you to one question each, please.
Thank you for the talk. I really like talks that present new directions or perspectives. So my question is kind of also on that and kind of covering the other half of the story that you didn't necessarily focus on, which is the cell plasticity. Because my own background is I work with cell epigenetic reprogramming, and we have to work very hard to get a cell to convert from one lineage to another, whether that's iPSC or transdifferentiation, right? And these are, in the same way, artificial transitions that don't occur in vivo. And what you've shown is, yes, this is an artificial transition we see with MSCs that doesn't occur in vivo. And so my question is exactly that. So why doesn't it occur? Why would you hypothesize it doesn't occur? And why would nature not take advantage of that plasticity and only use the immunomodulatory effects?
Well, I think the assumption that you're brilliant and that your in vitro experiments are mimics of what Mother Nature can do turns out to be, you may be brilliant, but that assumption is completely wrong. So there are very few systems that mimic an in vivo microenvironment. So the problem is you can't set up a mimetic of that. You can ask scientific questions in vitro. There are no animal models, none, that mimic disease states. There's animal models that we use to ask scientific questions. So the mistake is to think that what you see on a petri dish, all of the plasticity that we use to make those cells dance are sledgehammers. They're not little tinkles to the cell to make the cell response. They're massive changing microenvironments. So the answer to your question is, if you put those sledgehammers inside the body, you'd kill cells. So you can't see that plasticity in vivo, and don't worry about Mother Nature. She's much smarter than you and I, and she uses all of the clues and information to her advantage. But plasticity of mesenchymal cells is a cell culture artifact.
But it seems very unique because other cell types you get the culture conditions wrong.
I can't hear you.
I was saying it seems very unique because if you get the culture conditions wrong for other cell types, they just die, right? In this case,
No, again, you're not. You have to listen to what I said.
Yeah.
In vitro
Right.
is not a mimetic for in vivo.
Sure.
And so therefore you can futz around with the culture. We could take a skin cell and we could make it into an embryonic stem cell. We can take a skin cell and make it into an embryonic stem cell. That's a low, it turns out what the boys don't tell you in their papers is that it's a 1 in 10 million event. So it's not that we're so smart and know, even though some guy got a Nobel Prize for doing it, okay? It's a fabulous achievement, but it ain't happening on a mass basis, and therefore we can't use it, nor can Mother Nature.
Dr. Kaplan, I don't envy your evening because everyone's going to be inundating you with questions, and you probably won't get to eat. In the interest of time, we're going to move on with our program. Thank you. Thank you. Our next speaker is somebody you've been waiting for a long time, and I think everyone has this interest today is what is our regulatory status? Dr. Ken Berger has been incredibly busy in this field since 1888, I understand. And Watson and Crick days, yes. Not only is he interested as a hobby in regulatory affairs, but it's his part-time job. His full-time job is a soccer player. So I think he's been contracted with AC Milan in recent years, but an avid...
but they're not doing very well.
And he's probably going to compete and break that 2-hour marathon as well. So I'm going to let you have the...
Several times. It's like 4 hours for me.
Habib, are you jealous? So, Dr. Ken Berger, and I'm going to set up your PowerPoint.
Okay. Thank you. I'll be brief. I know it's lunchtime, and I don't want to give a long and complicated talk about regulatory, but it's quite obvious that whether it's a drugstore or it's a stem cell, they're drugs. They're biological drugs, and anything you do with them in the body is going to require FDA pre-approval. That hasn't really sunk into the Kaplan cell community that we have here. They are really under strict regulation, but only under moratorium as we sit here. One more year of, where am I? I don't want to sit down here.
You want us to do that?
Yeah, that's okay. So we have one more year of a moratorium that was set up by the prior commissioner. And when we say discretionary with FDA, it really means that. I've been in the regulatory part of pharmaceutical development for years, and FDA discretion can change in one day. What Gottlieb said could be changed by Sharpless, the new commissioner, or the new interim commissioner, and could be changed again by whoever is appointed as commissioner in the future. But knowing this, we have probably a year to get our act together and to develop investigational new drug applications for the clinical testing of these products, whatever you call them by any name, are biological drugs, clinical products that have to be tested. So, yeah.
Ken, you're just in the view, if you can step over this way.
I can do it at Kaplan and just stand down here, take a look at everybody and see if you're falling asleep yet. The reason why I'm not going to give a long talk is people do fall asleep. And so I wanted to give salient features such that I want you all to hear loud and clear that what you are working on and with are biological drugs, and they require an approval from the Food and Drug Administration. Having said that, it's going to be much easier for us because the FDA has no clue how to regulate us. They've come up with 4 guidances. They've come up with definitions of homology. If you're a blood relative, you can take the product without any approval. If you're a third removed blood relative, you have to go through a whole biological license application to use that product. So they really don't understand it, but they're in charge. So my recommendation to Habib's company has been and will be, let's get some INDs together. First and foremost, thanks to Dr. Kaplan, I have to change my placebo-controlled study. We have to do a run-in. That's a great clinical trial word. We have to do a run-in of saline for some time to see if these responders respond to saline. Now, I've done that before with other clinical trials. It's important to do because sometimes the device that you put in to deliver the drug has its own effect. This is true with shunts in the brain. So it's important to do a run-in, and thanks to Dr. Kaplan, I screwed up a clinical trial for Habib's company, Invitrx. So what we're looking for is intra-articular injection of Kaplan cells from the umbilical blood, and we'll test to see if pain can be reduced. We know that they're not going to make new cartilage, but we do know that pain is a big part of what we want to ameliorate in all these patients. And we know it can do a good job anecdotally, but as I met people last night and today, everyone says the same thing. Where are the clinical trials, placebo-controlled, randomized trials that prove this? And there's not a lot out there, but everyone has a poster child who they have helped and saved, and people get off from the gurneys and they can walk. But where is the proof? It has to come from us. It has to come from this group. Now, the FDA posed a scenario where real-time evidence could be used by consortia of physicians to generate, yeah, this is okay, I don't need this, to generate data. Well, I had a criticism of that, but so many people have written in and saying, What does that mean? It has to come from a capitalist system. Companies need to put money into it. How do these groups of physicians then market a product? Where is the centralized location of that consortium of doctors? Well, I could ask all of you to run these studies, but it really wouldn't hold up to a randomized trial, blinded. It just won't happen. So I have no idea. I like those guys. I have no idea what they were talking about. It's the head of the Center for Drugs and the commissioner. If anyone's read that paper, New England Journal of Medicine, if they can explain it to me, terrific. But I know this, that we're going to be better than conventional 10 years, $10 billion for cell therapy, because the public won't stand for it. The public didn't stand for it a couple of years ago. People are getting Kaplan cells infused, and they're getting support from their pain and other conditions. So just to go back to regulatory, and I'm only going to go for a few minutes. These are biological drugs, make no mistake. And if for the people who like codified information, it is not Section 361 alone. But don't get caught up in the nomenclature. It is Section 361 of the Public Health Service Act, as well as Section 351, which means you have a biological drug that has to be pre-approved by the FDA twice. First, they approve it to go into clinical trial, and then they approve it for commercial use. They may bypass, once you get an IND, and I've told this to Invitrx, they may allow you to commercialize because you already are. My advice to companies that are making these products is at least listen to what Gottlieb said. You have a moratorium until November of 2020 to at least submit an IND to go into clinical trial. They didn't say they're going to shut everybody down, but they said if you have not shown evidence that you're willing to work with the FDA, you could be in real trouble. Now, Congress, in August, directly asked Sharpless, the acting commissioner of FDA, okay, buddy, what are you going to do in November 2020? What's your plan? How many of these businesses are you going to shut down, if any? What is your plan? Now, I didn't have time last night to Google what Sharpless's response was, but his response may not be the same as the real commissioner who's about to be appointed. So you need to take all this with a grain of salt. But the one thing that I explained to Invitrx, and one thing that you all should remember in your practices, is whether you're a medical doctor or a chiropractor or whatever, the FDA still controls the delivery and labeling of drugs. You may feel they have nothing to do with the practice of medicine. The FDA has gone on record to say we could, but we take discretionary non-moves with doctors because they have medical boards and other people that they have other kings and queens to deal with. However, it's true that the FDA could do something. So all doctors, all people, all practitioners, pay attention to what's going to happen over the next year. It's very important. And the one thing that Congress knows and the FDA knows is that very few companies selling Kaplan cells in the United States and the world have gone to the FDA. Invitrx did. We submitted a request for an IND, meeting with them. That puts us on the radar screen. They know we're a good actor. Anybody else that's using product or anything, I would suggest you at least put yourself on record that you're working with a company working with the FDA. That's almost a sales pitch, isn't it? Well, it sounded good to me. So just in conclusion, please, the take-home message is these are drugs. They're biologics or drugs, and I don't know if they're Center for Drugs or Center for Biologics. Milad said they were Center for Biologics, but his slide did say Center for Drugs on it, so maybe that's just a typo. But usually what happens with these kind of products, like exosomes or the cells that are cell therapy, is both centers get involved because it's too complicated. You're dealing with proteins, which are usually Center for Drugs. You're dealing with cells, which are usually Center for Biologics. So I would say it's really an academic question, but you can bet both centers, ego centers they are, will get involved in the prosecution of the clinical trials for cell therapy. I have to say one thing, the Case Western and Yale, Case Western. Does anybody know the name Guido Cervalas? He was dean of Yale Law School. He was the guy who wrote the essay. The legal essay is, do we own our own body? There are many ways to say we don't. Conscription into the armed forces. Can we sell our heart? Do we own our heart? No. What happened at Case Western was very exciting because this was a Yale professor, dean, who went to Case Western to open up the question of, do we own our own body or our cells? Bone marrow patient. He needed bone marrow. His cousin had the right, whatever it is, match. I'm not going to give you my bone marrow because I'm afraid that I might die because I need my own bone marrow, not knowing that he could probably do this very safely. The patient died. Now I'm going to pose a question that I know Dr. Kaplan may think about in the next few days or year, because someone asked him the question, are we going to have donor-specific exciting cells? And does Habib have a right to keep his beautifully curtained, evasive cells? That's going to become a real issue in our society because there will be special donors that have special molecules that are life-saving. And if you think about it, the United States and the democratic system does not sit well, as some other countries do, where they will force people to donate their special property. So I like getting into philosophy because I took philosophy as a minor in college, but it's just something we should all think about. And many of you have some special cells. You do, and special exosomes. Are you going to sell them for $1 million? What's going to happen to the future? Well, I think these are not chemicals. These are not Center for Drug Chemicals. These are interesting avenues to analyze, to look for, and then to move forward on it. So as we move to allogeneic, as we move away from autologous, we're going to have to understand the master cell banks and who owns them. Is it going to be government controlled because companies fight against each other? What's going to happen? Because the future of medicine is here. We just heard it. You're going to go to an emergency room and you're going to get a bag of whose cells? Habib's? He'll be a billionaire, a trillionaire. What's going to happen economically, which is the cover for a drug approval? So the take-home message, we're drugs, biological drugs, we have to go through clinical trial. But it should open up as soon as you get an IND, and we're hoping this. I'm going to ask the FDA this question. Can we now then set that our GMP program is working? We have safe cells, and we have a myriad of indications that the doctor should be allowed to now practice. Exactly what we're doing now. We're in a GMP facility. We're providing safe cells or safe exosomes, and the doctor should be allowed to now prescribe them and use them in the field. That may be a game changer for the FDA. It will not be like it looks like this year, however. And everyone has to conform to the ego centers in Washington. Have to do it. And thank goodness that Habib listened to me a couple of years ago, and now we have a very good facility, and we can move forward with our INDs. INDs is plural because Milad wants the exosomes to go forward. We have a dentist somewhere in here who wants injections orally, which go to the rest of the body, right? So this is the future. I love what's happening. I knew Dr. Kaplan would give us many new things to think about. But on top of that, if we have donor selection, who's going to protect Habib if he's the donor? Thank you.
Thank you, Ken. Dr. Kaplan, this is a Saturday. You guys have given us way too much to think about, so it made me hungry. Just a little anecdote or a little humor here. Dr. Kaplan's correct. We as clinicians, we think we know everything about the human body, and I was a mouse mammary carcinoma researcher in the past, and I took care of a mouse colony, so I thought I knew animals. So one day when my cat fell a little behind on his tick and flea medicine, I said, hell, you know what? I'm just going to give him the dog medicine. And cat went into seizures. And so I said, wow, seizures, let me give him some Valium. I know everything about the body. And I rushed into the cat to the vet, and the vet just cracked up laughing. He says, you surgeons always think that you know it all. And he said, the cats and dogs are completely different biology, completely different anatomy, and the human drugs like Valium don't work on cats. And so it took about a week to recover my cat with special medication. But anyway, we're all hungry. We are staying on time, fortunately. I'll try to continue to keep you on time. We will have a session at the end of the day where all the panel will be available to ask questions. And I think we'll actually mic that if you want to, so that everybody can hear the question and answers during your cocktails. I promise you that around 4:30. And I'll even cut my talk short as well to accommodate that. So please go enjoy lunch right now. It'll be in exactly an hour, and I'll call you back to order then. Thank you so much.